Okay, sweet. Recording is started. So this, I'm probably not going to involve people that are here a whole lot just because, this is all new material. This is material that you all have not seen before. But first things first, I kind of want to give a little bit of a breakdown and a walkthrough of Phys 1 specifically. So I'll take a quick step back. Try 2. I did kind of already talk about this before. I have a video as well on my YouTube channel that kind of goes over um, like an intro to try 2. Let me pull that up so I can show that as well, so you can see what I'm talking about. Try 1 is difficult, typically for the reason that you're new to grad school. There's a lot going on, and it can be super, super frustrating because just like getting used to figuring out kind of what the heck is how the heck to do grad school, right? Try 2 is very, very much an info heavy. It's more difficult because it's very, very info heavy. Like there's a lot more potentially difficult material, especially if you're in person. I guess in person and online. There's a little bit more lab heavy. So you start to work in kind of the lab type stuff, and you work in just a little bit more information kind of in general is kind of what I felt. So there's a video here that kind of goes over each class, roughly what to expect. This one is a little bit outdated, just because when I recorded this, the clinical imaging courses, I didn't know a lot about them, as well as the teacher has changed for Phys 1. And the reason why I wanted to start now with Phys 1 is that is typically one of the biggest classes that holds people back. So try 1, it's typically biochem. Try 2, it's typically, again, either biochem 2 or Phys 1, and occasionally as well gross anatomy 1. Those are kind of the 3 just very heavy science courses that there's a lot of information in there and a lot to kind of remember and figure out and really study and review in that course. So kind of what we're going to do, and kind of I can show you guys this. What I'm going to focus more on is actually the previous professor's notes that I learned the material off of. Mostly because the difference in this material, I should say the difference in the slides of the previous professor has a lot more material, a lot more information than the current professor. For some reason, not even for some reason, physiology is a really, really difficult class to teach in general. Difficult to teach and difficult to understand because there's a lot of kind of what I would call circular understanding, where in order to understand point A, you need to understand point B. And in order to understand point B, you need to understand point C. In order to understand point C, you have to understand point D. And in order to understand point D, you have to understand point A. So it's kind of like this wheel of trying to figure things out, especially in the beginning, when there's just a lot of overlap between all of the different material. Um, it's easiest and best to kind of break things apart, do each of the different things separately, learn each chunk separately, and then put all of the pieces back together. Additionally, kind of what I've heard, the current professor is Dr. Tate, Kevin Tate. from what I've heard, he is good, like a really, really cool person, not the most organized or linear teaching individual. Can, like with live lecture, he can jump around a lot, kind of say one thing and then backtrack and then say another thing. So it can be a little bit difficult to follow. And that's one of the big reasons why I'm jumping back in to do physiology tutoring. Also, if you are on campus, there is another tutor. I'm not sure what his hours are. I think he does Monday nights, large group sessions, named Cameron Mahoney. He is, no, sorry, Carson Mahoney. He's super, super cool. He's a good friend of mine. So if you want more of like if you are in person, you want more of like an in-person kind of thing, or kind of go to both of these and absorb more information, he's a really good resource as well to kind of to balance out. Biggest piece of advice I'm going to say, I just say this with a lot of courses, but especially this trimester, physiology is very, very, very difficult to catch back up in. If you fall behind at the beginning, you're basically kind of shafted. It's not impossible to get back into things, but if you dig yourself into a hole at the very beginning of the trimester with this course, it's a very difficult hole to crawl yourself out of. So that's why I want to get started. I wanted to get started early and kind of started. And a big part of that is because of what the previous teacher would call the try to hangover. When you have your first break, let's see, this first break, we start school back on the 6th, technically. So we have these 2 whole weeks, and technically finals kind of ended on Thursday, so we had a little bit more time. So kind of 2.5 weeks where, in all honesty, a lot of people are just going to kind of do nothing. If you're watching this as a recording, very, very good. Try and keep up and start looking at this stuff early, because if you get out of a rhythm, it's going to be, yeah, Kristen, you tried to hangover last try. Yeah, it's very, very real. Where as soon as the first 2 weeks of the trimester, because try one, you could kind of take the first 2 or 3 weeks to like figure out what the heck is going on and then kind of buckle down and get things going. Try two leaves no room for that. You need to hit the ground at a full sprint and just start grinding through right from the beginning. So if you're not on some kind of set schedule, getting up at a certain time, going to bed at a certain time, still kind of, going back, kind of reviewing, prepping for this next try, staying in some sort of feeling of a schedule, of kind of a set, solid schedule, get back onto that. If not this week, next week, before the try starts, get into something of a schedule, because trust me, if you don't, it's going to be very, very difficult. Before we jump into a bunch of stuff, does anybody have any specific concerns, questions, anything about the course in general that either you've heard or that you want clarified right now before we keep going?
No, I would honestly just say more that I'm kind of nervous about this class because I took this class in undergrad, and you are definitely right. It's not linear learning, unfortunately. And I managed a C, but the way that the questions were being asked, it got me really confused because there can be multiple right answers in physiology, but one best answer. And I feel like that's just kind of harder to grasp in a sense. So, and I don't know if there's Linus Learning on this, but is there a way that there's like questions that we could do or resources like that, like with biochem?
I don't believe that there's not a Linus Learning per se. I'm fairly certain. I'll have to double check. that Dr. Tate does have weekly like online questions and stuff. I'm gonna probably need to reach out to him and get his permission to be able to kind of go over some of those things, and maybe potentially use like his online resources on online quizzes and stuff, to do similar kind of what we're doing with Linus Learning questions. If not, there are some other resources I can find. I can pull and find other questions as well, so around physiology, to do something similar to this. I'll reach out to him. Let me actually do that right now. I'm going to message Dr. Tate. I've chatted with him a couple times. I'm going to message him and see if he has something um, that either he can share with me or something like that. For the most part, from what I've heard, his tests are difficult, but the test questions themselves and the quiz questions, at least the ones that I've seen especially, have been very fair. Like if you know and understand the material in the way that I'm going to present it to you guys and kind of go through it with you, you'll be pretty good. And I'll give you the tips and the tricks and kind of the know-how and the ways to really internalize the material, especially because with physiology, everything builds on itself. So that's also why I say you can't fall behind, because if you don't fully understand, like this very first PowerPoint talks about the cell membrane, diffusion and osmosis, the different pumps and membrane potentials, all of this stuff. If you don't understand this now, like when we do, not like right now, but if you don't understand these points, before like your first test, as soon as you finish the first test, the second chunk of material is going to pull very heavily from this, as if you have like a really good understanding of this. So the course is very, very much going to build on itself. And I'm going to teach it the way that I'm going to kind of go over it with you guys is going to be very much focused, like doing that. So like I said, I'll try and do what I can with like getting practice questions and doing all that kind of stuff. So, and we'll kind of get there when we get there. Physiology is a little bit different because biochemistry, believe it or not, you didn't have as much lecture as you'll have with phys. Phys should you'll have two-hour sessions of lecture for phys 1. So it's 4 hours of lecture. So obviously we won't be able to go through all of the material together. With my schedule, I'm going to try my best to probably do a 2-hour session of phys, which we can get through a good chunk of stuff. I'll see if I can find another time to do more recordings. I have a couple videos up for at least for the first like 4 or 5 chapters. For the first exam, I have videos all up already on the YouTube channel. But then after that, I'll try and either keep recording or try and figure out another session, to get all of this material recorded so that you guys have a way to kind of supplement your learning. And I would recommend, it's my personal recommendation, go for all of your professors, go to their in-person lectures if you're able to. Go to their in-person lectures, or like the recordings or whatever they have, if they do live sessions. Go at least once or twice and see if like how well you can learn the material from those live sessions. If with any professor, if you feel like you do not get benefit from the live sessions, 2 choices. 1, go to the live session and sit in the very back of the room, throw your earbuds in, and work on other schoolwork, watch recordings of the live lectures, or find another way, either watch recordings after the fact or use other resources, my YouTube videos, Khan Academy, Ninja Nerd, all that kind of stuff, to understand and work through the material yourself. Like I said, this is very much a subject where the more time, you're going to have to spend some time with it. in order to get to know it. And the best way that I can describe it is physiology. Really, you just have to spend enough time with it to get to a point where something clicks. And as soon as you get that click, aha moment, a lot of the pieces will fall into place. If you don't get that, if you don't have that moment yet, you just need to spend more time with the material in order to get to that point. So those are going to be my recommendations. I know I've said a lot. Anything else? Burning hole question.
Kind of just a general class question in general, but I swear my previous classes I was able to find the syllabus before it was even available on Blackboard. But I don't remember where I found that. Do you know possibly where?
Let me see. Usually the syllabi aren't available until like the class is open. Typically. So I wouldn't know where to find that. But day one is going to be your whole syllabus day to do all of that. So sometimes you can find the syllabus if you go into, I think it's in My Parker. Let me see. I think within My Parker, you can search it. Sometimes those are not always updated to the most recent. But sometimes if you go to My Parker and you up at the very top, like here's My Parker, and you go up to the search and you look up like whatever the class like number is, and then search like the syllabus physiology 1. Sometimes it'll pop up here. Let's look. And also, if you haven't seen this, if you ever go into My Parker and like search stuff, they have a lot of materials on My Parker for like part 1 boards and stuff. Like here's a document. This one's horrible to read, but these are practice questions for part 1 boards of for physiology and that kind of stuff. So there are a lot of resources on My Parker if you haven't played with that. Additionally, if you haven't played with this either, the library resources, I didn't realize this was a thing until more recently, but you have on the library, you have databases and catalogs and journals. Here's a really, really cool one. This is recorded so people can watch this. You go to My Parker, databases, under O, official disability guideline. This is a database that's typically like, I think it's $600 or $700 a year that people pay for. And you can search for any of the any of the ICD-10 codes, diagnostic codings, like rotator cuff syndrome, and it'll take you to like different type of individual, as well as right here different scenarios. And typically, as you keep going out, it'll go through all of this stuff, as well as treatments, specifically giving you different treatments, and it'll show you different research, like what the research says is the best ways to do all this stuff. So a little off topic, but My Parker has a lot of cool stuff, and the library has a lot of cool stuff. So use your resources. Okay, there's no other questions. We're going to jump in to lesson 1, which is cell membranes. Last chance. Any last questions? Okay, and if anything does come up, feel free to shoot me a message or leave a comment on the video once it's posted. I get notified whenever people leave comments, and I can take a look at it, and either I can message you directly or answer comments and stuff there. So okay, so talking about physiology, this is kind of the overarching theme of physiology, and that is homeostasis. Specifically, homeostasis, the best way to describe homeostasis There's dynamic equilibrium is one definition, but super even simpler one is just balance. So the body needs to be balanced. There are very specific conditions within which each of the components and the parts of the body works, and it needs to remain within those parameters to properly function. So all physiology talks about is how the body maintains that balance with all of the different stressors that get placed on the body. A stressor can be referred to any sort of outside or internal force that's causing a change. So like if I put my hand on an ice block, that's an external stressor. If I start exercising, that's a stressor. If I all of a sudden see a bear and my sympathetic nervous system shoots up, seeing the bear is a stressor, and my body needs to balance and respond to that appropriately. So it's all about keeping our bodies in balance and keeping us alive with whatever we're trying to do. We do focus just a little bit. This is kind of like the general material. Talk about the cell membrane. Hopefully you remember a lot of this with the lipid bilayer made up of proteins and carbohydrates, that it's selectively permeable to water and different water-soluble molecules. When we talk about this especially, we're going to talk a lot about osmosis, diffusion, and the different types and styles, which is what we're going to get into in a little bit. But specifically, if we're ever talking about water, if you ever see water, like specifically H2O, and H2O is moving across a membrane, that is osmosis. If there's ever other stuff moving across a membrane, that's going to be some kind of diffusion or transport. Transport, diffusion, those two words can kind of be used interchangeably, but we'll get there when we get there. Okay. The mitochondria. Hopefully, after Biochem 1, y'all are sick of talking about the mitochondria and how we have that outer membrane, the inner membrane, the matrix, all of that sort of stuff. But the same thing, there is a lipid bilayer. These membranes are lipid bilayers, and that's the site of that aerobic respiration, which we'll get into. That's that Krebs cycle, all that sort of good stuff from Biochem 1. A lot of the nitty-gritty stuff from Biochem 1, not so much the nitty-gritty detail stuff, more of like the general concepts. A lot of those concepts with like metabolism and that kind of stuff are going to carry over into physiology. So you should start to see some things that you recognize. There's our metabolism. Typically, at least I know for sure, Dr. Tate, let me actually pull up, make sure that I pull up the Dr. Tate stuff. I know I got that shared, all of that shared with me recently, so I can kind of put it side by side and make sure that I don't miss anything that if he added anything. Last time I checked, it was all pretty much the same. Here. Let's take a second to pull up, but there it goes. Okay. Yeah, so a lot of this, he has it in here, kind of going over it. It's not, he doesn't, this is more of a review kind of stuff. It's not going to be it's heavily tested on or tested on at all. Just kind of here to remind you kind of what's going on. Here's the lipid bilayer. That lipid bilayer is a barrier to the water and the water soluble substances, but the other molecules can still move through it. So we have certain things, ions, glucose, H2O, and urea, which really can't get through it super well. H2O is semi-permeable. And then remember that there are other things like carbon dioxide, oxygen, certain specific ions in glucose that can was I muted for a while?
Only about 15, maybe 20 seconds.
Okay. I don't know how that happened. Okay. So lipid bilayer. Understand that this is, you're going to want to look and see kind of what things can pass through and what can't pass through the lipid bilayer. So certain ions and like big, larger ions or heavily charged ions can't pass through. Glucose can't pass through. We know that because we need that hexokinase and those GLUT transporters to bring things across. Gases like carbon dioxide, oxygen, nitrogen, halothane. They never ask about halothane, but like oxygen and carbon dioxide are big ones that they can pass freely through. Urea, H2O, glucose, and other ions can freely pass as well. So you'll see a handful of general questions about this sort of stuff. This is a really good thing to kind of add into a study guide and just honestly go through and Google kind of the most common things that are able to pass through the lipid bilayer. Membrane proteins. This is where we start to get into a lot of the important stuff that we really want to focus on. And we're going to kind of do a little bit of a circle around and around with this material specifically, because this is going to come back and forth. Okay, so we're going to jump around a little bit when we talk about this. Okay, so the membrane proteins, as it sounds, membrane proteins, these are proteins that are found within the membrane, and really a lot of the time these are our intermembrane proteins. If we think back to Biochem 1, especially towards the end of Biochem 1, can anybody give me an example of an intermembrane protein, say, when we talk about like the electron transport chain?
So like
You
the
mean like
complexes.
one?
So like
Yeah,
complex
the
one,
complexes.
complex 2.
Not complex 2, because remember that doesn't go all the way across, but complex 1, complex 3, complex 4, complex 5, right? Because those go from both sides of the membrane. And in this example we see here, this has potassium. When we looked at the complexes, it was hydrogen coming through. So exactly what we were talking about before, there's specific ions and specific things that these channels or these carrier proteins are going to allow to come and flow through, either from outside to inside the cell or from inside to outside the cell, depending on what the function of that protein is. We'll see this again a little bit later. I'm just going to mention it now. Anytime that we're using a protein of some sort, some kind of ion channel or carrier protein, which are both intermembranous proteins. These are both ion channels are made up of proteins and carrier proteins. Anytime we're using either of these, that's going to be called a facilitated type of diffusion. Okay, it's going to be a facilitated action. So we're facilitating. Facilitate means to kind of to help or to allow to happen. So the proteins and the protein channels are what are going to facilitate or allow that type of movement of ions to happen. So we talk about something called diffusion, and I'm going to put up Dr. Tate's material side by side. So as you can see, Dr. Tate's material, it's pretty to the point, but I felt like I had to break down a lot of stuff and move a lot of stuff around. So that's why I prefer the previous professor's material. But so when we talk about diffusion, we're going to be over here on the left. So diffusion is going to occur down a concentration gradient. Remember in Biochem 1, we talked about, again, electron transport chain, pumping those hydrogens across. We created that chemical gradient or that concentration gradient. Same thing, that just like it happened there, where once we pumped everything up using the energy from the electrons, then it flowed back through complex 5 with the ATP synthase in a direction of high concentration to low concentration. Okay. And diffusion only occurs if a cell membrane is permeable somehow. If there is no protein, just like we saw in that slide before, only certain things are allowed to pass through, kind of those non-polar molecules, some of those gases, that kind of stuff is allowed to pass through. Once we have a protein, it allows for more stuff to come across. He makes the specification that non-polar solutes are going to go, they're going to do simple diffusion. I call it simple diffusion, meaning that no protein is used to assist. And then if you have a polar solute or a polar type molecule, you're going to require facilitated transport or the use of some kind of protein. Okay, so that's diffusion. A little bit lower down here, we talked about osmosis. We mentioned that osmosis is very, very similar to diffusion. Main difference is it's talking about movement of water. Diffusion is the movement of like the molecules and the solutes and the stuff mixed into the liquid. So those proteins, those ions, the oxygen, carbon dioxide, all that stuff. Whereas osmosis is specifically the movement of water. And as well, this image right here, you guys recognize this stuff? Isotonic, hypotonic, and hypertonic. You guys recognize that?
Yes.
Yes.
Yeah, so this is going to come back again. Most of the professors for physiology, they like questions involving these different types of solution and kind of what's going to happen to the cell. So remember that with an isotonic solution, the concentration gradient, or like the concentrations comparing inside of the cell to the outside of the cell. So isotonic, anytime you see iso means same. You're going to see a lot of this prefix. So remember that iso means the same. Tonic. So isotonic solution means the concentration inside and outside of the cell is the same. If concentration is the same, there is no high side and low side. So water will not move. There's no changes to the cell whatsoever. Hypertonic concentration is high outside of the cell. So we have a bunch of stuff. It's kind of like we had a cell floating in regular water, and then we pour a bunch of salt into that water and mix it up. Now the water has a bunch of stuff inside of it. So we have a really, really high concentration outside of the cell, lower concentration inside of the cell. And when we're looking at this, our body wants to create homeostasis. So the cells want to do homeostasis. They want to have balance. So they want the concentration on the outside to equal the concentration on the inside. So the way that it's going to do that is move water. So with a hypertonic solution, water goes from water moves out of the cell and into the solution. So the cell shrinks. So because it's trying to dilute the surrounding, because this is all high concentration here, because it wants to dilute it, the water is going to come out. A hypotonic is the opposite of the hypertonic. Instead of so the concentration outside of the cell is super, super low compared to the inside. So water rushes in to try to balance it, and this you get swelling of the cell. The way that I always remember this, you really just need to remember one, hyper or hypo. I always remember hippo, because hippos are fat. So if it's a hypotonic solution, the cell gets fat. Okay, any questions about osmosis or diffusion or anything else that we've talked about up to this point? Hopefully it seems pretty straightforward right now.
No questions, just I wish I had the hippo thing a lot earlier in my life than now.
Yeah, you'll see you will not get rid of that isotonic solution question. These questions, these will pop up over and over and over and over again through the whole program. Like, I don't know why, but for some reason everybody loves these questions. I had 2 of these questions on my first board exam. Was the hypotonic solution type questions. A little bit more wordy, kind of like you give a person a saline with whatever percentage salinity, and compared to normal salinity of blood, which is this, what's going to happen to the cells? Like, that was the kind of question I got, but it's hypo, isotonic solution type questions. Teachers love this stuff. It's never going away. Okay. So that's osmosis. Always remember osmosis, water. If you ever have to pick an answer choice, if you're stuck between answer choices and you have osmosis, osmosis is being either in the question or in the answer choice. Osmosis always pairs with water, okay? And diffusion can be with any of the other stuff. So now we get into what's called passive versus active transport. Okay. Let me find, there it is in here. We'll start with passive. There should be, let's see if there's a designated slide for passive, because, okay. We'll use this slide, but I'm going to also build another kind of little connecting chart, because kind of similar to biochemistry. You know how in biochemistry, a lot of the different terminology had AKAs of each other? Very, very similar with physiology, where there can be a lot of different words that all mean the exact same thing. So we're going to cover a couple of those, okay? So we talk about passive transport. We talked about diffusion. We talk about passive transport. Passive transport. All passive transport is saying is ions are transported, ions or other stuff other than water. Because if it's water, what do we call that? What's it called when we move water across a membrane?
Osmosis.
Osmosis, good. So anytime we move ions or other stuff other than water are transported without the use of energy being ATP, that is passive transport, okay? So no matter what is happening, if we are not using ATP, no matter how things move or cross membranes or do whatever, if no energy is being used, it's passive transport. Okay, biggest thing to look at if you ever have an answer choice that says passive, you better be darn sure that no ATP is involved anywhere. And that should make the other side of the coin, active transport, a little bit easier. Requires energy. 99% of the time, this is in the form of ATP. Couple small exceptions is like when we were doing electron transport chain stuff. With the electron transport chain, we used the energy from those electrons in order to move hydrogens. But for pretty much, I'm fairly confident, 95% confident that everything we're going to talk about within physiology 1, we're going to be saying if it uses specifically ATP, it's active transport. Okay, that's what we're going to look for. Always look to see if ATP is used. Now, backing up a little bit, I mentioned that word facilitated, right? Facilitated diffusion / transport. Does anybody remember what I said about facilitated diffusion or facilitated transport? What is required to be present in order for it to be considered facilitated?
Proteins.
Some kind of intermembrane protein, whether that be in the form of an ion channel or a, what was the other word that they use? Here, carrier protein. Ion channel or carrier protein, that is going to be facilitated. Now, so these are kind of separated by this line. And they're their own categories, but we can stack them on top of each other. For example, our sodium, potassium, ATP pump, or sodium potassium pump. Do you guys remember? Remember hearing about this? I think it was mentioned briefly in Cells and Tissues and a little bit in biochemistry here and there.
Yes.
Remember that? Do you remember any of the job or the function of that sodium potassium pump?
It was to change the membrane potential of the cell, right?
Yeah, specifically its job is to maintain cell membrane potential. Help maintain that cell membrane potential. The way that it does that, and I guarantee you this is a test question, it pumps 3 sodium out of the cell and brings in 2 potassium. Another super important thing to remember about this, and we'll see some of the AKAs here. Where's the... And you'll see a couple of names. Sodium potassium pump. It can be written as sodium potassium ATPase. It can be written as sodium potassium pump. Anything like this, you'll see. Okay, so it pumps the 3 sodium out, brings 2 potassium, in. This is a test question. It's always a test question all the time. And because it is an ATPase, it breaks down ATP and uses ATP to drive this reaction or this process, whatever you want to call it. So we look at this and there's 2 parts to this. Let me actually pull up a picture of the sodium potassium pump. Here's our sodium potassium pump, just like this. He uses ATP, pumps out the 3 sodium, brings in the 2 potassium, just like we talked about. That right there is an intermembrane protein. So that means it is facilitated. And then it uses ATP. So if it uses ATP, what kind of transport do we say that is?
Active transport.
Active. So we can combine all of these and call this, it is an active facilitated transport, just like that. So these different categories can kind of be mixed and matched and placed together. So just like we have an active facilitated transport, we can also have a passive facilitated transport. There's something that we'll learn later on down the road when we get to the heart. That's in chapter 4. It's right before the first exam. That's I believe it's week 4 is when you'll learn about these. There's something that they call funny channels. These are sodium ion channels that allow for sodium to slowly leak in order for the heart to beat on its own without the brain telling it to. That's called autorhythmicity. That's further on down the road, just introducing you to that concept now. But you can have both of these. You can have the active facilitated transport. You can have passive facilitated transport. When you go to answer test questions, you don't always have to have all of these words, but you want to remember. So for example, if I were to ask you the question, the sodium potassium pump represents which of the following? And I gave you the answer choices: simple diffusion, passive facilitated transport, active facilitated transport, and active transport. Technically, this is where sometimes teacher like physiology can get really confusing, where there are technically 2 correct answers, but we want the best answer. So while active transport technically is a correct answer, the active facilitated transport is the best answer of them. So Dr. Tate, from what I've seen, doesn't do a ton of this, but you will kind of see it here and there throughout, just because of the way physiology is and kind of the way that the material all links together. Okay. Any questions about all of this stuff, about passive, active, facilitated, the examples and stuff that we used? Any questions about any of that? Sweet. Again, if you have any questions, feel free to speak up. Okay. If I remember, I don't believe Dr. Tate talks about any of this stuff. If he does ever mention this kind of thing, there are these things called aquaporins. They're water channels. So they're just protein channels. This is an example of it's facilitated, but it's also passive because it doesn't require ATP. Water molecules can just pass through however they want. So aquaporins are an example of passive facilitated diffusion or transport. Cool. Another thing to note, kind of talking about the cell membrane itself, things that are lipid soluble molecules are able to just naturally cross the phospholipid bilayer. So look out for that kind of stuff. Again, like I said, I would make a note somewhere when you get to that first week of material, what things can cross the membrane, what things cannot cross the membrane, why for both of those, that kind of stuff. A lot of times that knowing that, it will come very much in handy in understanding things that happen in the future. Diffusion versus osmosis. We talked about this. Remember, osmosis is movement of the water. Diffusion is movement of the molecules and stuff that are mixed into the water, the solutes. Okay. Ion channels. So now we're going to kind of break down a little bit further. Wrong document here. Facilitated diffusion and transport. We have these ion channels and we have the carrier proteins. We're going to look deeper at the ion channels because there are different kinds of ion channels that do different jobs and react differently. So we have ungated channels and then we have gated channels. So the ungated channels, these guys are just going to sit and be open all of the time. For example, like the aquaporins, these are considered ungated channels because there's not a gate stopping things from coming through. What determines if something is able to pass through the ion channel are things like their size, their shape, or like the electrical charge of either of the channel and the ion. So kind of like how positive and how two positives and two negatives repel each other. If you have a negatively charged ion channel, typically it'll be some kind of positively charged ion that is able to pass through that channel. An example of this are those leaky ion channels, just like we talked about. We call them funny channels in the heart. Again, you'll talk about those in the future. We'll get to those later on because they do play a very important role within the heart. But those are ungated channels. Okay, they're always going to be open. It's just the size of that ion, size, shape, and charge that determine whether or not it's able to pass through. Then we have gated channels. So let me actually do this. So ion channels. So we're going to go right here. Actually, let's do this. And honestly, this kind of format, this is how I would do a lot of my notes for this course because we're going to be jumping back and forth. So ion channels, we have gated. We have ungated. Yeah. Ungated and gated ion channels. Ungated, always open. Depends on the ion and its characteristics if it can pass through. Our gated channels, we have two subcategories of gated channels. We have voltage gated and chemical or ligand gated. We've mentioned ligands before in biochemistry. Does anybody remember what a ligand is? If not, that's fine.
Like a substrate?
Yeah, it's some kind of substrate, and specifically a substrate that's going to go and bind onto some kind of protein, right? It's that chemical messenger that's going to go and bind onto an active site. So we saw that with kind of like remember the G protein coupled receptors from Biochem 1? Whatever would come and bind to those G protein coupled receptors, that was a ligand. So we're going to see the same thing here. We have ion channels that are going to respond to specific chemicals. Most commonly, the one that we're going to look at is acetylcholine, which is one of our chemical mediators. So yeah, we'll look a little bit more into that in a sec. Okay, so looking here, let me just double check Dr. Tate's stuff and make sure that we've talked about all of this. So we talked about simple diffusion, we talked about facilitated, we talked about active, and this is why I prefer to use the previous professor stuff, because he'll just throw a picture in here, and I feel like there's a lot of extra stuff. He says a lot in lecture, but again, if you miss some of that, it can be difficult to go back and relearn this stuff. The leaky channels, aka the ungated channels. Again, these are all AKAs. So if you see leaky or ungated, either of those, that's the same thing. And then he also throws in here mechanoreceptor gated channels. So let's add that to our list here as well. Mechanoreceptor gated. Okay. So when we talk about voltage gated, we're going to get more into the voltage gated and all that kind of stuff. Again, this is one of those things where what we talked about in the beginning, kind of to understand A, you have to understand the D and the C and the B, and where it kind of loops around. So we'll come back to these voltage gated channels. What you need to know right now is that all of our cells have what's called a resting membrane potential. So for example, for neurons, it's negative 70 millivolts. This is the typical charge, like the difference between inside and outside of the cell. The inside of the cell overall is more negative. Specifically for nerve cells, it's negative 70. For certain nerve cells, it's negative 70, the main ones that we'll focus on. And so this charge of negative 70 is going to be changed due to some kind of ion channel, typically a ligand gated channel, that will cause it to depolarize. So it will become more positive till it gets to a point where the voltage gated channels then open and allow for the rest of rapid depolarization. That's a lot of words, a lot of things that are kind of all over the place. We're going to come back to these types of channels and kind of what they mean later on and look at that whole process in detail. So right now, don't worry too much about this. I just want to introduce them. The ligand gated channels, so chemical messengers bind to active sites and cause them to open. Big one that we'll talk about is the acetylcholine receptor channels. These are also going to be called the nicotinic. We're going to loop back later on. We see like those nicotinic, muscarinic channels. We touched on those in Biochem 1. And I always hated learning all of those. So we're going to circle back to those in the future to kind of look at each of those and understand the naming of all of those. Mechanoreceptor gated, we'll get to these even a little more. This will be kind of the next chunk of PowerPoints when we talk about specifically with like smooth muscle activation kind of stuff. This is where an actual physical movement, like a movement, like the movement, either like the pressing onto a tissue or movement of tissue in some kind of in some way, is going to cause receptors and channels to open. So for example, like if you press onto your skin and then you feel that sensation, that's because when you press, the mechanoreceptors, at your nerve endings that are on your skin, when you press, that causes those to open. That force causes them to open, which releases a signal into your sensory nerves, which travels up into your spinal cord, into your brain, and tells your brain that you're feeling something right there. Those are what mechanoreceptor gated channels are responsible for. Okay. I believe that's as deep as Dr. Tate typically goes. Yeah. Okay. We'll get into the membrane potential stuff now. Dr. Van Winkle, who was a previous professor, goes a little bit deeper into this stuff. If you want to look at all this and use all this stuff again, like the link to fill out the order form for my notes and everything is in the description of the YouTube video, or you can message me for that and check it out. Like I said, there's a lot more information in all of her slides. Some of it Dr. Tate might mention in class. Some of it might just be kind of extra to help you understand what's going on. So here we talk about like the rate of diffusion, how things go, electrical potential. Same thing as talking about membrane potential. We looked at this a little bit. We have a voltmeter, which measures the voltage difference between the two, how the outside of the cells, specifically, we're going to look at nerve cells. Nerve cells, we're going to focus, this whole first chunk of material is going to be looking at the nerve cells. We see they have, it's technically like between negative 65 and like negative 75, something like that. We typically call it negative 70. If for whatever reason, Dr. Tate or whatever professor calls it something different, I always go with negative 70 millivolts because it's easier for me to remember the straight number when you start learning other stuff as well. Okay. Let me see if he ever talks about the nerds. He does. It's a little bit down. Okay, we'll get to that when we get to that. Okay. Again, Van Winkle stuff, this goes a little bit deeper into all of this, the gradients and diffusion and all that kind of stuff. Dr. Tate doesn't go into this. I'm just going to leave this slide here for a second. If he ever does mention something like this, you guys can come back to and look at this, or just Google primary versus secondary active transport and kind of the way that works. I haven't seen anything with Dr. Tate that he teaches this. It's not super important or necessary to understand the future material. So there's the sodium potassium pump. Something you'll notice about Van Winkle's material is you'll see a lot of repetitive slides. So slides saying pretty much the exact same information, just in different ways or different photos, that kind of stuff. She was very, very thorough in her PowerPoints of giving people multiple kind of ways of understanding stuff. Okay. We'll look at memory potential. with the membrane potentials, we have our what's called our resting membrane potential, which is just that. So when nothing is happening to the cell, it sits at its resting membrane potential voltage. So for nerves, again, that's that -70. When we start to go more positive, we get something that's called depolarization. So the cell gets more positive. There's another term called repolarization, where cell goes back negative. Hyperpolarization is when the cell is more negative than the resting membrane potential. A lot of this is just definition type stuff. This will come back again in the future when we start looking at what's something that's called the action potential. So what actually happens within that cell to help us transmit a message and a signal. Okay, so these are just definitions. Depolarization, repolarization, hyperpolarization. You will very, very much need to remember these definitions. What contributes to the membrane potential? So the cell permeability and ion concentration affect the membrane potential. The different ions are unique. Main thing that we want to remember here is that inside of the cell, you have very, very high potassium, which is positive, and all of our amino acids, not all, but most of our amino acids and our proteins are very highly negative. So we have high potassium, which is positive, and high amino acids or proteins, which is very, very negative. So that's where we get that the overall charge inside of the cell. So because of the amino acids / proteins inside the cell, the inside is highly negative. And then on the outside, we have lots of sodium ions as well as high chloride ions, but way more sodium ions. So it's thanks to all of the sodium that we have a positive outside, which then the positive outside, high negative inside, that's where we get that membrane potential from when we measure it. Positive on the outside, more negative on the inside. This is saying all of that. Same thing. This gets a little bit into kind of what's called chemical and electrical forces, where if we think back to what we're talking about, about diffusion, as well as kind of electronegativity, where positives and negatives attract each other. If something is positively charged, it wants to go towards the negatively charged thing. Or if it's positive, it wants to go away from other positively charged stuff. So you get these forces, a chemical force, meaning that because there's a bunch of potassium on the inside and not a lot of potassium on the outside, potassium would like to go out of the cell to be away from all the other high concentration of potassium. But also that the electrical force for potassium, because the inside of the cell is negative, that pulls the potassium inwards. And then for sodium, has both of them going in. This is why it's easier for sodium to go into the cell than it is for potassium to just leave the cell. Kind of jumped around a little bit there. Is everybody feeling okay up to this point? Any questions, comments, concerns? Has what I has what I have talked about so far up to this point, has it all made okay sense, more or less? Or are there parts that are confusing?
Makes sense.
Makes sense. Yeah.
Okay.
Yeah.
Good. Uh, okay. So this right here, when we talk about, again, that balance, we reach what's called an equilibrium point, where we have overall sodium wants to come in, potassium does want to leave more than it wants to stay in. So we do get this occurring. We have potassium coming in, or sorry, sodium coming in, we have potassium leaving, but it reaches a kind of a steady, with the sodium potassium pump, with the help of that, we get kind of a steady state develops. So we have a slight, overall, we have a slight inflow of sodium and an outflow of potassium. And then the sodium potassium pump, the sodium potassium ATPase, is also doing its job at the same time to all work together to create this steady state, or this balanced point, also called an equilibrium point, which is that resting membrane potential, and it keeps it around -70 millivolts, specifically for nervous cells. And we can actually look here on Dr. Van Winkle's PowerPoints. Different cells have different resting potentials. Neurons, we could go with -70. Smooth muscle fibers, typically, we can do either -50 or -60. Skeletal muscle fibers, we typically say they're around -90. And those are the different resting potentials. This is very similar to biochemistry, where we had a couple of equations that, more than anything, it was like, hey, just remember kind of what the equation is and more or less what it tells us. So the Nernst equation, it solves for the membrane potential for a single ion. So for example, looks at how much potassium is on the inside, how much potassium is on the outside, and we get a membrane potential, so a charge comparing those two to tell us what that is. So for example, just specifically for potassium, we'd plug in the different ions, like on the outside, the concentration on the outside versus the concentration on the inside, and that would give us that membrane potential for the single ion. You don't need to know how to do all of the math. You'll just want to be able to recognize the equation and know this simple definition, that it gives us the membrane potential for a single ion. Then, same thing here, we have what's called the Goldman-Hodgkin-Katz equation. This just takes the Nernst equation and solves it for all of the ions that are around, for multiple ions. So it includes the potassium and the sodium and the chloride, and you can add more and more ions, and that will give you the value of the overall membrane permeability and membrane potential for all of this stuff combined. Again, recognize the equation, recognize what it does. So the question that you'll get one of these, and it says, which of the following equations represents the membrane potential for multiple ions? And it will look something like this. And this is the one you just want to remember, the 61 log of whatever, remembering that type of equation. Don't worry about, I haven't heard anything about him or any other professor making you do the math on this stuff. So don't worry about that. This isn't like a math heavy program. So we said that sodium, potassium, ATPase maintains that resting membrane potential. The net negative charge on that inside. We use the ATP, so it's that active facilitated transport. All that good stuff. Okay. See if we missed anything here. So again, here on this side, we're looking again, those ligand channels, the voltage gated, the leak channels. This goes more over, just use a lot more repetitive slides. Sodium, potassium ATPase, kind of what simple diffusion looks like through the leaky channels. We looked at depolarization, hyperpolarization, repolarization. Other definitions, excitability, and threshold. We'll look at these again in a little bit. Here we have, again, the gated channels, mechanical, chemical, and voltage. Repeating a little bit here. He mentions Ohm's law. Again, this is going to be recognize the name and roughly what it tells us. Nothing should be super math related. Okay. Then we get to really, so a lot of what we just did is laying the groundwork, important kind of material to understand now what is action potentials. How's everybody doing up at this point? Are we feeling okay?
Doing all
We
right.
chilling.
We chilling. Okay. This next portion, this is the meat and potatoes of this kind of lesson. What's really, really, really crucial to understand and to know going forward. Action potentials is something that's going to be brought up over and over and over and over and over again through a good chunk of all of exam 1, a good chunk of exam 2, and you will see, I promise you'll see this stuff as well on the final exam. So this is the kind of stuff we really want to know this, because this is not going to go away at all. Okay. So specifically, when we say the word action potential, so the action potential is literally, it's called an excitation coming from the neuron, where you have rapid rise and a subsequent fall in voltage across a cellular membrane. The change in that electrical potential, so that change in that rise and the fall of that voltage, is associated with the passage of an impulse along the membrane of a muscle cell or a nerve cell. So action potentials, in simpler words, these are the actual messages our body sends along the nerves or the muscles to communicate what needs or what is happening. Okay, so it's with action potentials are what carry all of our messages throughout our bodies. They're literally, this is how nerves work. This is what we're going to talk about, is how our nerves actually send messages and communicate messages from our body parts up to our brain, and from our brain back to out to our body. It's connecting all of us together. So the action potential, it's used for long distance communication. And this is key right here. I guarantee this is a test question as well. An action potential is an all or nothing response, meaning either we have an action potential that's going to send a message, and it's that message is going to go and go and go and go for whatever it needs, wherever it needs to go to, or it's not going to happen at all. Okay? There is no kind of like, oh, there's a baby action potential or there's a really big action potential. No. An action potential is always going to be the same size. Specifically, like if we're looking at a graph, it's always going to be the same size. It's never going to, get smaller, and drop off and go away. Once it starts, it goes all the way to its destination, unless there's some kind of like pathology or something that prevents it or stops it from doing that. Okay, and we're not really talking about or dealing with pathology right now. So we're just going to assume for now, either it happens or it does not. Okay. And I am going to stop Dr. Tate's slides and switch over to the other ones because these ones do a much, much better, clear job. All of this. So this is what our action potential looks like. I highly, highly recommend. Actually, I'm literally going to tell you, do this. Print off like 6 of these. Okay, or more. Or be able to learn how to draw it specifically from memory. Something like, let's go, like this one right here. This is perfect. Starts at -70 up to a little bit less than +40. All this, all through here. Knowing what is happening at every single step of this guy is going to give you a ton of your answers to questions on exam 1 and during the first 2 weeks of the course. Okay? Super, super, super important. And we're going to walk through step by step exactly what is happening throughout this. So this right here, this is the example of an action potential for a neuron. And let's label a couple of things to start with. So this value right here, -70. Can anybody tell me what that value, -70, represents? What did we say?
Resting membrane potential.
Yeah, the resting membrane potential. What's the name of the protein or the protein that is responsible for maintaining the resting membrane potential?
The sodium potassium pump.
Correct. Yeah, the sodium potassium pump. Also known as the sodium potassium ATPase. Either one. That sodium potassium pump maintains that resting membrane potential. So what happens, we get some kind of stimulus to our body. A lot of the times this is going to be some kind of, it can be either mechanical or it can be a ligand or something like that. A lot of the times we're going to say a good chunk of the time there's being mechanical. It doesn't necessarily matter exactly what that stimulus is, but that stimulus is occurring right here. We have some kind of stimulus. Whether that be like we're touched or like our brain wants us to move, some kind of chemical gets released to activate something at the very beginning. Whatever that is, doesn't matter. We have some kind of stimulus that's going to begin the process. Something acts on a nerve cell. Okay? When it acts on that nerve cell, let me make sure I use the exact words. Dr. Tate uses this graph. Okay. Dr. Tate, in his thing, he specifically calls it a depolarizing stimulus. So this stimulus is going to cause some slow depolarization of the cell. What's key is that we're looking to hit this dashed line right here that's known as the threshold. Okay? This is what determines if we have an action potential occur or not. If we do not meet threshold, no action potential occurs. If we do meet threshold, an action potential happens. And that's that all or nothing. Okay? So let's say we have a stimulus that starts to depolarize our neuron, our nervous cell, and it reaches threshold. As soon as we reach threshold right here, we have voltage gated sodium channels begin to open. So, and then when you have this printed off and you start to draw this, I want you to write it just like this. So the voltage gated sodium begin to open right there. Okay. And then we get depolarization. So that all this is saying, we get depolarization of our nerve cell. Depolarization is when it goes more positive. So we have a whole bunch of sodium rushing in because we're getting depolarized. Okay, that's that rapid sodium entry depolarizes the cell. As we get up here towards the top, as after we go past 0 right here, at this point, those voltage gated sodium channels start to close. Sorry, the writing is really gross, but it's fine. So here, those voltage gated sodium channels start to close. At the very, very tip top right here, as soon as we hit the very tip top, the sodium channels are closed. Okay, so they start to close as we get up close to here. And once we get all the way up, they are officially closed. At this point, as well, right around here at this point, voltage in this same area here as well, voltage gated potassium channels start to open. So remember when we talked earlier about where sodium wanted to go and where potassium wanted to go? Sodium wants to go into the cell. Where does potassium want to go to?
Out.
Wants to go out. We have a bunch of potassium on the inside. Potassium overall wants to go out and leave the cell. So as soon as that voltage gated potassium channel opens, so it starts to open here, and once it's finished opening here, we get a whole bunch of potassium. So in this one, we have potassium leaving the cell, and we get opposite of depolarization, we get repolarization. And it goes. As soon as we get down past our threshold, those voltage gated potassium channels start to close. They're a little bit slow to close. So the voltage gated potassium channels take a little bit of time to close. So you get this extra little dip down, and that is called hyperpolarization. Hyper-repolarization, sorry. Hyper-repolarization. So we go a little bit more negative. And this has a very, very crucial job right here. The reason why we have this hyperpolarization This is called our refractory period. Let me find the good picture. So here we have, this is why I like Dr. Van Winkle's notes. She has a lot of very, very detailed notes of like exactly what is happening, where, and when. I want to find the picture with the here we go. This is a slightly different this is a different type of cell, but the concept is still the same. When the cell, as soon as we start depolarizing, we get a different color. As soon as we pass threshold and till here, we have what's called an absolute refractory period. What that means is that no matter what happens, no matter what is going on, we cannot have another action potential happen. Because the channels, those voltage gated channels and everything, they're already open, they're already doing their job. There's no way that you can reopen them when like we're way up here, when our voltage is way up here. There's no way in that area that we can reopen or reclose these kind of things because they're dependent on the voltage. So the absolute refractory period, there's no way that we can have another action potential occur. After that, so here, once we've returned, we have what's called a relative refractory period, which means we're not super likely to have another action potential happen because we're hyperpolarized. But if we have a big enough stimulus, a big enough depolarizing stimulus, we can force this to get up to reach threshold and cause another action potential to occur. Okay, so this is more definition type stuff for you guys there. Again, the easiest way to remember all of this, I highly, highly, highly recommend Ninja Nerd on this. So right here, Ninja Nerd has a great video. The only issue with Ninja Nerd videos are that they are pretty long. But if you watch them, you will have most of the material that you need for pretty much any of this stuff. So he does a great, great video going over a lot of this stuff that we talked about, creative potential, all of this, how it balances out, how he gets a little bit more in depth, a little bit more technical. So right in here, he goes over all of this, and I think he draws it all out like this. I don't think I think he covers he covers the graph at right here towards the end. So if you want to go and watch the end of this video, this starts at about 45 minutes. Yeah, about 45 minutes. So if you watch from the 45-minute mark, exactly what we just did with that graph, he's going to redo it. He'll do it right there. And he does a great job at a lot of this material. So, but this right here, this is going to be key to a whole bunch of questions that you will get asked about threshold, what channels are opening when, and how they're opening. based because of like the voltages, what is happening when these gates, specific gates, are open or closed, et cetera, et cetera, et cetera, all of that kind of information. So very, very important to learn, draw this out a handful of times. I would get a copy. I have my copy somewhere. I'll have to see if I can find it, where I printed this out and I wrote it very, very detailed, very, very neatly, kind of like as a guide, as a key that I could come back to and look at. Okay. I think for now, because we've already talked about a good chunk of stuff, let's end on this. Next time that we meet up and talk, we'll dive a little bit more into these gates themselves and how they work. They have different components that are opening and closing, where we'll add more detail to this specific graph about what's happening, even more detailed within each of these different gates, with each of these different gated ion channels. Before we wrap up, any questions, comments, concerns?
None right now, at least.
Okay. And biggest thing to remember is you guys are ahead of the curve right now. This is like, you're not learning any of this. The big goal of this is, to get you guys to see this material, at least a good chunk of this material. Hopefully we'll get through at least like the first week and a half's worth of material, hopefully maybe 2 weeks worth of material before we get into, you guys actually get into the try. So that way, while the panic ensues of a new trimester, you'll already have a decent foundation built as you get in and start to see this material.
And then you'll let me know. I went ahead and was making like an outlined Word doc as we were going over stuff. So if y'all want it, let me know. I'll send it to you.
Yes, please.
Okay.
Also, yes, please.
As well, once we get into the actual try, so like I said, this class can be a little bit trickier, whereas like with biochemistry, we could typically like go through the whole Linus learning thing just in one tutoring session. Phys is going to have more lecture, and like realistically, like we want to spend a little bit more time going through stuff. It's going to have more lecture than probably what we're going to have time to go through. So we'll kind of play with it as the try goes along. Whether or not we stick to doing one session, if we try some weeks to do 2 sessions, like I said, I'll try and do more like practice question-y type stuff as well. Because that also helps us kind of cover important material a little bit faster. But yeah, we'll kind of play with it. It'll help a lot if, as you guys go through material, and then once the try actually starts and you go through those first couple of lectures, write down specific questions or concerns or things that are confusing that you want to go over here in tutoring. This will be a much smaller group compared to your actual class. So it'll be much easier, to kind of really dig into the nitty gritty confusing stuff with less people here than it will be in your course.